last part of our lecture on Mendelian genetics, we're going to go beyond traditional Mendelian genetics and look at some other types of genetics. Not every single gene out there has one dominant and one recessive allele associated with it. In fact, a lot of genes out there have more than two alleles associated with them, or those alleles don't behave in a strictly dominant recessive fashion. So we're going to look at a couple examples of these. Complete dominance, which is the type of dominance that Mendel studied, is not the only kind of dominance out there. There are actually other types of dominance, including a type of dominance called incomplete dominance. And in incomplete dominance, the dominant allele is not able to completely cover over or mask the effects, the phenotypic effects of the recessive allele. And I'll give you an example. If I have a, this is a hibiscus flower. If I have a red hibiscus flower and I cross it with a white hibiscus flower, the coloration here is incomplete dominant. So the combination of the two, the red is not able to completely cover over the effects of the white allele, and as a result we get flowers that have a pinkish appearance to them. They're kind of like the average between the red and the white flower. This is a little like that blended inheritance theory I mentioned earlier. It's a little bit like that. Anyway, incomplete dominance again just means the dominant allele isn't absolutely dominant. It's only partially dominant. I'll give you a couple of examples in humans. Curly versus straight hair. If you have an allele for curly hair and an allele for straight hair, instead of just having curly hair or having just straight hair, you will have wavy hair, which is sort of halfway in between curly and straight. That's an example of incomplete dominance. Another example is sickle cell anemia. It's a disease I mentioned in a previous lecture to you. Sickle cell anemia is where you have red blood cells that contain hemoglobin that is not completely functional, and it leads to all kinds of serious health problems as a result. A person that has one copy of the normal allele for making hemoglobin and one copy of the recessive sickle cell anemia allele will have a moderate version of the disease. They won't have a full-blown version of sickle cell anemia because they have some ability to manufacture normal hemoglobin. So we say they have sickle cell trait rather than the full anemia disease. Another kind of dominance is something called codominance. Codominance, don't confuse it with incomplete dominance. In codominance, you have two dominant alleles, and they both are fully phenotypically expressed in the offspring. The best example I can give of this is blood type. You're all familiar with blood type. People can be type A, their blood can be type B, their blood can be type O, their blood can be type AB. Type A blood is a dominant blood type, okay? And we write that as I sub A, capital I sub A for type A blood. Type B blood is also a dominant blood type allele. And and then type O, by the way, is a recessive allele, so I write it as little I sub O. If I were to unite a gamete with a type A allele and a gamete with a type B allele, the two of them together would be type AI, type B. Both of these types would be fully expressed in the offspring. We would have type AB blood. If this were like incomplete dominance, you would have something that was the average between A and B. Maybe it would be like type C blood or something, which isn't the case at all. Both of these alleles are fully expressed phenotypically. It's not like one partially covers over the effects of the other as an incomplete dominance. Here, both alleles are completely and fully expressed in the phenotype. This is codominance. Interestingly enough, blood types also exemplify another feature that we haven't seen before, and that is a multiple allele system. Rather than just having a single dominant trait and a single recessive trait, in the blood type system that we're familiar with, the ABO blood typing, there's a dominant A allele, a dominant B allele, and a recessive O allele. So this is an example of a multiple allele system. Now, a lot of times, one gene does not influence a single trait. A lot of times, a number of genes interact with each other in order to produce the final phenotype that we see in a person. Skin color is a good example of that. There's actually 3 different genes involved that interact with each other, and the combination of alleles you get from those 3 genes interact to determine whether you have extremely light skin or whether your skin is darker. Human height is similarly affected by multiple genes, as is eye color. We think there's at least 5 and possibly more genes involved in determining the color of your eye. The color of the seeds in wheat, the plant wheat, also involves a polygenic inheritance scheme. Polygenic inheritance means more than one gene influences the trait. You remember again, poly means many. Genic relates to genes, so this is many genes influence the trait. One other thing, we can have the reverse happen. This is a process called epistasis. In epistasis, one gene affects multiple traits. Okay, before we had, on the last page there, we had several genes, gene 1, gene 2, gene 3, all contribute together to determine one trait. Now we're doing the reverse. Now we have one gene affecting many traits. A good example of this is a human disease called Marfan syndrome. And this is a disease that historians believe that the President Abraham Lincoln had. In Marfan disease, you have individuals with this disease, one of their genes that codes for the manufacture of a type of collagen found inside cells. Collagen is a type of fibrous protein. They have a defective gene for the manufacture of that collagen protein. Collagen happens to be a protein that's found in places like the heart, the muscles, the eyes, the cartilage, bones. It's found throughout the body, so having an inability to properly make that collagen means that the people usually have vision problems. They usually die of heart attacks at a much younger age because they have a weakened heart. Their fingers and toes tend to be extremely long because the bones and ligaments have been affected by the defective collagen gene. They tend to have a chest that's concave instead of convex in the sternum. So they have lots of issues and problems with many different parts of the body as a result of one defective gene. This is called epistasis. Last thing, and I'm only going to touch on this briefly, even though there's an entire field of science devoted to this and tons of research going on in this area. Genetics aren't the end-all be-all. The environment in which we place ourselves can influence our genotype and ultimately then influence the resulting phenotype. A great example of this is what happens in these cute little Himalayan rabbits. Here's a pretty Himalayan rabbit here. The coloration of the fur in Himalayan rabbits is determined by certain genes. However, those genes can be turned on and off dependent upon the environmental circumstances that rabbit is experiencing. So for example, you can do a research experiment, and this has been done, where you strap little ice packs to the back of bunnies, poor little bunnies running around with ice packs on their back, and after leaving those ice packs on for a good amount of time till new fur can grow, the fur that grows in its place will be of a different color. Now if you think about Himalayan rabbits, if they're in an area where it's really cold, the best way to warm up is to absorb sunshine, the warmth from the sun, and the best way to absorb heat from the sun is to have darker fur. So obviously this would be an advantage to be able to change fur color dependent on the environmental conditions. So this is an example where environment interplays with your genotype to determine the phenotype. There's actually a whole field of science right now that's just coming on board called epigenetics, and this is looking at precisely how it is that the environment interplays with our genetic makeup to change our genotype even within the course of change our phenotype within the course of our lifetime. And it can even cause impacts and effects on things like the gametes we make and thus the types of offspring we have as a result. But that's that could be an entire class, an entire semester's worth of study. We're not going to go into that. I just wanted to make mention of it here.